Hello, I'm Bob Prue, CEO of Imagion Biosystems. We're pleased to be a sponsor once again of the VBIC's annual event and are grateful to the conference organizers for the opportunity to present to you today our work in developing magnetic nanoparticles as imaging agents. There are multiple forms of magnetic imaging technologies, both commercially available and in development. Dr. Alt from Monash Biomedical Imaging Group has already covered how superparamagnetic nanoparticles are used in the newly emerging field of magnetic particle imaging. And I expect many of you are already familiar with their use as contrast agents with magnetic resonance imaging, but may not yet be familiar with the technique of magnetic relaxometry. This is an imaging modality that looks at the relaxation rate of magnetic nanoparticles. When nanoparticles become bound or attached to tissue, they relax more slowly from a magnetized state. Using ultrasensitive detectors, we can differentiate rapid relaxation by Brownian motion from the slower nail relaxation that occurs when the particles aren't free to rotate. This slower relaxation results in a detectable magnetic dipole within the field of view. You can think of magnetic relaxometry as a form of hotspot detection, like with PET tracers, but with magnetic moments detected instead of radioactivity. Now, there's been a long history of using magnetic nanoparticles as contrast agent for MRI, and there are certain advantages compared to conventional gadolinium-based contrast agents, including long circulating half-life and lower risks of toxicity. But today in the US and most major markets, there is no approved iron oxide-based imaging agent. This then results in the product ferraheme, which is approved for the treatment of anemia, being used off-label when looking to use an alternative to gadolinium. The 2017 review paper shown here provides an excellent overview of the potential clinical utility of iron oxide-based imaging. But with all that clinical work, it begs the question, why haven't iron oxide nanoparticles been brought to market for use with MRI? I think there are three contributing factors. Firstly, contrast agents have been regulated as drugs, making it expensive to develop relative to the value for reimbursement. Secondly, the image contrast generated is negative or dark, which is the opposite of what most radiologists are used to for image enhancement. And thirdly, the products made to date have been non-targeted agents and not designed as fit for purpose. So in the end, what we have seen is clinical evidence that they provide limited incremental improvement over conventional imaging, are not in alignment with users, and are expensive to bring to market. Let's look at an example. In the study shown here, the iron oxide agent Residus was administered to breast cancer patients to see if it would aid in determining nodal status. The idea being that the non-targeted dextran-coated nanoparticles would be taken up by macrophages. With this approach, lymph nodes that appear as dark are node negative, while nodes that show no change in signal intensity are node positive. In the top panel, you can see from the left to right a CT identifying a sentinel node, followed by a pre-dose MRI and a post-dose MRI. In the post-dose MR, signal intensity was low, signifying SPIO enhancement. In the bottom panel, you see the same sequence, but in this patient, pre-dose and post-dose signal intensity is similar, indicating there's no SPIO enhancement. The conclusion here being that the lack of contrast agent indicates that the macrophages have been displaced by tumor cells. And while this is an improvement over using imaging to assess morphology and identify suspicious nodes, it still doesn't provide confirmatory evidence that what is in the node is tumor. So what we've been working on is going from using magnetic particles for imaging enhancement to using particles for true molecular imaging. We do that by starting with an iron oxide core and biofunctionalizing it with antibodies to target delivery to the tumor phenotype being assessed. One of the great benefits of this targeted imaging agent approach is that we can change the antibody for targeting different tumors without having to reinvent the underlying construct. PET tracers, for example, have to be carefully matched for their half-life with the targeting moiety, but the underlying construct with magnetic nanoparticles doesn't change. While 25 nanometer pores are ideal for our magnetic relaxometry and seem to be suitable for MPI, we know there may be other applications where different size may be beneficial. In developing the MagSense imaging agent, we have developed a method for producing the iron oxide pores to very tight specifications and high quality, which we think will translate into improved imaging agents. At present, our first targeted magnetic particle as an imaging agent is being used in an early feasibility phase 1 study in Australia with 4 hospitals participating. Like the example provided earlier, the idea is to see if nodal staging can be accomplished non-invasively, but now with a targeted imaging agent. Our MagSense HER2 agent is designed to specifically bind to HER2-expressing tumor cells. For the study, newly diagnosed HER2 breast cancer patients are given a pre-dose MRI, followed by a subcutaneous injection in the breast of the imaging agent, and MR scans at 24 hours and 72 hours post-injection. A biopsy of one or more of the nodes provides a tissue sample, which is then measured ex vivo with our magnetic relaxometry system to determine if the nodes exhibit a magnetic dipole associated with the bound nanoparticles. Biopsied samples will also have pathology assessment to compare the 2 imaging modalities, MRI and relaxometry, against the standard of care. Here I'd like to acknowledge the support we received from Siemens in Australia, who worked closely with the radiology team at the Austin to optimize the MR protocols we are using in the study. To date, multiple patients have been enrolled and have completed the course of the study. We aim to complete a minimum of 12 to 15 patients, but may leave the study open further to gather additional information to inform the development of the product for a larger pivotal study. We're excited to be working with our Australian clinical researchers on this first of its kind study. We hope to demonstrate that targeted magnetic nanoparticles are a biosafe alternative, enabling true molecular imaging where the imaging agent not only improves image contrast, but confirms the presence of tumor cells. Thanks for your attention, and I hope you have a successful event.